Gene therapy is an experimental technique that uses genes to treat or prevent disease. In the future, this technique may allow doctors to treat a disorder by inserting a gene into a patient's cells instead of using drugs or surgery. Researchers are testing several approaches to gene therapy, including replacing a mutated gene that causes disease with a healthy copy of the gene, inactivating, or knocking out, a mutated gene that is functioning improperly, introducing a new gene into the body to help fight a disease. Although gene therapy is a promising treatment option for a number of diseases, including inherited disorders, some types of cancer, and certain viral infections, the technique remains risky and is still under study to make sure that it will be safe and effective. Gene therapy is currently being tested only for diseases that have no other cures. Gene therapy is designed to introduce genetic material into cells to compensate for abnormal genes or to make a beneficial protein. If a mutated gene causes a necessary protein to be faulty or missing, gene therapy may be able to introduce a normal copy of the gene to restore the function of the protein. A gene that is inserted directly into a cell usually does not function. Instead, a carrier called a vector is genetically engineered to deliver the gene. Certain viruses are often used as vectors because they can deliver the new gene by infecting the cell. The viruses are modified so they can't cause disease when used in people. Some types of virus, such as retroviruses, integrate their genetic material, including the new gene, into a chromosome in the human cell. Other viruses, such as adenoviruses, introduce their DNA into the nucleus of the cell, but the DNA is not integrated into a chromosome. The vector can be injected or given intravenously, by IV, directly into a specific tissue in the body, where it is taken up by individual cells. Alternately, a sample of the patient's cells can be removed and exposed to the vector in a laboratory setting. The cells containing the vector are then returned to the patient. If the treatment is successful, the new gene delivered by the vector will make a functioning protein. Researchers must overcome many technical challenges before gene therapy will be a practical approach to treating disease. For example, scientists must find better ways to deliver genes and target them to particular cells. They must also ensure that new genes are precisely controlled by the body. Gene therapy replaces a faulty gene or adds a new gene in an attempt to cure disease or improve your body's ability to fight disease. Gene therapy holds promise for treating a wide range of diseases, such as cancer, cystic fibrosis, heart disease, diabetes, hemophilia, and AIDS. Researchers are still studying how and when to use gene therapy. Currently, in the United States, gene therapy is available only as part of a clinical trial. In somatic gene therapy, the therapeutic genes are transferred into the somatic cells, non-sex cells, or body, of a patient. Any modifications and effects will be restricted to the individual patient only, and will not be inherited by the patient's offspring or later generations. Somatic gene therapy represents the mainstream line of current basic and clinical research, where the therapeutic DNA transgene, either integrated in the genome or as an external apisome or plasmid, is used to treat a disease in an individual. Somatic cell gene therapy involves the placement of a human gene into a living person's somatic cells that do not produce the eggs and sperm that in turn produce the next generation. Somatic cell gene therapy would aim to cure a disease only in the patient, not in the patient's descendants. It was initially conceived as introducing a properly functioning copy of a gene into a person who had a genetic disease as a result of inheriting only improperly functioning copies. Different types of somatic cell gene therapy have since been investigated for the treatment of diseases that are not primarily caused by inherited genes, such as AIDS and cancer. Over 100 clinical trials of somatic cell gene therapy have taken place. Very few have, thus far, shown any success. In germline gene therapy, germ cells, sperm or eggs, are modified by the introduction of functional genes, which are integrated into their genomes. Germ cells will combine to form a zygote, which will divide to produce all the other cells in an organism, and therefore if a germ cell is genetically modified, then all the cells in the organism will contain the modified gene. Germline gene therapy is the replacement of genes in which an offspring would inherit a new trait. This type of gene modification is still in the early stages of human medical intervention, but examples do exist, such as in cows modified to have elevated levels of milk production or the capability to secrete human hormones, or knock-in and knockout mouse models, used for decades to elucidate gene function. Germline gene therapy targets the reproductive cells, meaning any changes made to the DNA will be passed on to the next generation. Germline gene therapy is when DNA is transferred into the cells that produce reproductive cells, eggs or sperm, in the body. This type of therapy allows for the correction of disease-causing gene variants that are certain to be passed down from generation to generation. Presently, there are a number of strategies that are mainly used in somatic cells, such as gene addition, gene correction, gene silencing, reprogramming, cell elimination. Germ cells are not used because of ethical issues. By using germ cells, it is theoretically possible to create a perfect human being. Thank you for listening. 1K likes please.